Hi everyone. So I would like to start today by talking to you about aging. Many people say that they feel a lot younger than their chronological age. And for some of us, this may actually be true. We know that there's individual variability in the biological aging process. This is why, if you compare two 60-year-olds, you may find that one of them is starting to suffer from the ailments of older age, such as joint pains or even arthritis, while the other one is still running marathons. And one of the key factors in this discrepancy is a person's telomere length. Telomeres are repetitive nucleotide sequences that are located at the tips of our chromosomes and that don't carry any genetic information, but that are there to protect our DNA. Very simplified, they can be explained through an analogy to shoelaces that have these little plastic caps sitting at the end of them, which protects the fabric from unraveling. Now, if you imagine that the fabric of the lace represents the DNA strands in our chromosome, then the telomeric nucleotides are wrapped around the DNA fabric at the end to cluster together like a protective cap, like the plastic cap, to then protect our DNA. Now, because our DNA is not replicated to 100% during mitosis, telomeres shorten a little bit during each cell division. And this is exactly their job, to act like a buffer zone so we don't actually lose any DNA information during mitosis. And because of this, also older age is correlated with shorter telomeres. Once the telomeres reach a critical length, the signal receives the cell to stop dividing, and it then enters senescence, at which point the functions of the cell will slowly become impaired. And because of this mechanism, shorter telomeres are also associated with an earlier onset of aging-related diseases and even with mortality. You can here see evidence from a study where the people with the shortest telomeres had the highest risk of suffering from cancer-related mortality. But it is also this last point which makes telomeres so interesting and why they have gained so much scientific attention, because it suggests that if we could maintain our telomeres or even lengthen them, our cells could continue to renew themselves and we could thus extend our lifespan, or at least the amount of time we can spend living without aging-related diseases. Aging-related telomere attrition is thought to occur slowly and gradually at a rate of about 30 base pairs per year, and it's normally measured after an interval of a couple of years. But there's now some very recent evidence that telomeres may actually change a lot quicker on an individual level after just a couple of months. And this includes evidence from intervention studies where participants underwent rigorous exercise training and after 6 months displayed significantly lengthened telomeres. But this evidence is also controversially discussed, particularly the evidence of lengthening, because it implies the reversal of a biological aging process, and we just don't know if that's possible. Further, since we know that in the long run, telomeres gradually shorten on average, it's completely unclear what these short-term changes really mean. Do they really represent a shift in a person's aging trajectory towards accelerated or decelerated aging? Or are they maybe something that just happens to the telomeres in isolation that doesn't have any further biological significance? And if the short-term changes are biologically relevant, then what are the more specific implications? Cross-sectional telomere length has been associated with health and aging very generally, but it's possible that these short-term changes have more specific implications, for example, for normal versus abnormal aging trajectories. And we approach these questions in a very intuitive way, particularly first point. If short-term changes in telomere length do represent a more generally biologically meaningful process, then they're very unlikely to happen in isolation, but rather they should be linked to other markers of health and aging. And this is now finally where neuroanatomy comes in. Brain structure, or in this case cortical thickness, can of course be considered to be a general marker of aging and also of neurological health. So we wanted to know whether short-term changes in telomere length were also associated with changes in cortical thickness. And to address this question, we collected data from 67 middle-aged adults. We acquired repeated measures of blood samples for the calculation of telomere length, which is done in leukocytes, and also acquired multiple T1-weighted MRI images. The data acquisition occurred at 4 time points, here T0 to T3, and they were spaced approximately at 3-month intervals. For the calculation of cortical thickness, we normalized each subject's cortical thickness scores at each vertex, and then calculated cortical thickness change maps by calculating vertex-wise difference scores. And finally, we assessed the potential association between change in telomere length and change in cortical thickness on a whole brain level through a linear mixed model that controlled for the influence of age, BMI, and sex. And what we found was that change in telomere length was indeed associated with change in cortical thickness, and part of the precuneus extending to the posterior cingulate. The scatter plot here shows the average change in cortical thickness in the identified cluster, plotted against the change in telomere length. It's LTL for leukocyte telomere length, and it's D, not delta, because we corrected the telomere length for regression to the mean, which is the gold standard. So one of the strengths of our studies is, of course, that we covered multiple intervals of change. So this enabled us to assess the consistency of this association across the 3 change intervals. To this end, we conducted cluster-level analyses for the separate intervals of change, and we found significant associations at the first 2 intervals and a sort of trend-level association at the final interval, which suggests that the association is relatively consistent. The next thing we noticed was that we have similar observations, or similar amount of observations, of telomere shortening and telomere lengthening. And as you will remember, it's the lengthening that is particularly controversial. So we wanted to know whether the instances of shortening and lengthening each contributed to the observed association to a similar degree, or whether maybe the association was primarily driven by the shortening in telomeres, which could then suggest that the measured telomere lengthening is a less reliable phenomenon or something that happens more in isolation to the telomeres. Again, we conducted cluster-level analyses, and we here found that shortening of telomeres and lengthening of telomeres were both associated to change in cortical thickness. In other words, the shortening of telomeres was associated with a greater tendency towards cortical thinning, and the lengthening of telomeres to a greater tendency for cortical thickening. So this suggests that the association that we find spans both degenerative and also restorative processes. So what does this all mean with regard to our initial questions? Well, our evidence suggests that the short-term changes in telomere length do indeed reflect a more general biological process that also affects other markers of aging, in this case, cortical thickness. And this is, of course, a very promising message for the previous findings of telomere lengthening, because it suggests that these training interventions that these studies did could actually lead to a meaningful shift in a person's aging trajectory. We can, however, say very little about the actual origin of the detected association. Further, with regard to the more specific implications of short-term telomere length change, we can maybe generate some hypotheses based on the identified region. The precuneus and posterior cingulates and their structure have been associated with telomere length cross-sectionally before, and we are here the first to show that change in these 2 markers is also associated. The precuneus is, of course, a very interesting structure that we've heard a lot about at this conference. It's one of the most highly metabolic active regions of the brain, and many of you will know it as the central node of the default mode network. But it's also associated with the development of aging-related diseases, most notably maybe Alzheimer's disease. And interestingly, shorter telomeres have also been associated with Alzheimer's disease and other aging-related diseases. So if the short-term changes in telomere length are particularly tightly coupled with the precuneus and posterior cingulate, this could potentially suggest that they're particularly meaningful for abnormal aging trajectories versus normal aging trajectories. But this is really quite speculative, and we of course need future work that actually tracks these long-term health outcomes to make any causal connections. So with that, I would like to close by inviting you to join me at my poster later today. And I hope that I sparked some interest in telomeres, because we've seen some really excellent and fascinating work about the connection between DNA information and brain structure already at this conference, and I would love to see telomeres more widely represented in that in the future. Thanks also to all of my collaborators at the Independent Research Group of Social Stress and Family Health at the MPI in Leipzig, and thank you to OHBM for inviting me today.